Thank you very much. I'd like to thank you for the kind invitation to come and speak to this, the most important part of the meeting, the patients and the individuals who actually suffer with this condition. And we researchers just need to remember that we're only here on your behalf to try and do things to help you and ultimately to cure this disease. So I'd like to start off with my declarations of interest. Thank you, John. So my declarations of interest all relate to GlaxoSmithKline, who I've worked with in a variety of capacities for many years. And in particular, over the past 5 or 6 years, we've worked together to try and develop small molecules that ultimately will cure this disease. And that's what I want to talk about today, if I may. So a bit of background about me, as John said in the introduction. For 22 years, I worked in a small place in the Fens called Cambridge. This is my house. And then 2.5 years ago, I realized this was no longer big enough for me, and I traded up and moved to London. This is actually Buckingham Palace. It's not my laboratories, but I aspired when I went there to have something a bit like this. And I have a research group and a clinic that still focuses on antitrypsin deficiency. So you recognize more than anyone that this is a genetic disorder, and it's characterized by this substitution in a protein. It's a substitution of a negative amino acid for a positive amino acid. You can think of it as a necklace, and one of those beads on the necklace changes from negative to positive, which means that when you put it together, it doesn't fold properly and hence causes disease. It's very common. It's more common than people think, and it's often underdiagnosed. As you all recognize, in individuals with 2 abnormal copies of the gene, 2 ZZs, then antitrypsin levels are low because the protein doesn't escape from the liver. And this accumulation of protein within the liver causes liver disease, and this is a liver of a 3-year-old boy who we transplanted for antitrypsin deficiency. And you can see here a healthy liver for comparison. So this liver is shrunken, scarred, you can see the nodules, see the areas of fibrosis compared with the healthy adult liver. If we take this liver, and actually I can tell you that 20 years ago I stood outside the operating theatre as a young research fellow and collected this liver as it was heading towards the bin and then took it back to the lab. You'll notice that there was no informed consent and there was no ethics approval, because why would you need it? Here was a bit of dead liver that was going in the bin. I couldn't do that nowadays, and I'd get into terrible trouble. But in those days, it was fair game, so I collected this liver and took it back to the laboratory. And if you section this liver, you can see that the mutant protein, the disease protein, accumulates within the cell. And here it is. Here's the amorphous abnormal antitrypsin inside this cell, causes the cell to distend and ultimately to die. And 23 years ago, I purified this protein from the liver and showed that it wasn't random aggregates of antitrypsin, but actually was made up of chains of polymers. And these polymers have a characteristic beads on a string appearance. So each white blob here is an antitrypsin molecule, and you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 as a beautiful chain of polymers. And what happens in individuals who are Z homozygotes is the abnormal protein forms chains of polymers, and these polymers tangle within the liver and they can't escape. And that's the cause of disease. So for me, this is a disease of polymerization. And what you want to know is exactly how one molecule links with its neighbor. So to summarize a huge amount of work, solving the crystal structures of the protein, looking at intermediates, looking at pathways of polymerization, this is how I think the protein links together. Here's the antitrypsin molecule. You can see the Z mutation sits here. It opens the molecule to form an intermediate that we called M star. Another molecule links together, and hence you have 2 molecules, and that extends to 3, 4, 5, 6 to form the chains of polymers. Let me show you that in another illustration. Here the molecules are colored red or white. It doesn't matter, they're all the same. But you can see the red molecule linking with the white molecule, linking with the red, and so on to form the chains of polymers. And that's what causes disease. Let me simplify it further. Antitrypsin effectively functions as a mousetrap. And what happens in this mousetrap is that it sets a bait that inhibits toxic enzymes. The mousetrap is held by a plus and a minus amino acid. If you change that one to a plus, you have a plus and a plus. They repel. The trap isn't held so stably. So in the liver, when the protein folds, all the mousetraps are firing. What does that do? There we have, at very high resolution, or very low resolution, that's the structure of the polymers, linked together as a series of mousetraps. And these mousetraps account for this chain of polymers that we find within liver cells. But as I've said to you, this is a disease of polymers. We find polymers in the liver, and so I spent a lifetime trying to find polymers elsewhere in individuals who are ZZs. And what we did is to take 240 ZZ individuals with the help of colleagues in the US, and we said, can we find polymers in your circulation? Can we measure polymers in the circulation of individuals? And what we found is that every single individual who is a Z homozygote has polymers in the circulation. Everyone. And what we could find is that there was a very weak association between the level of polymers and COPD. So for me, having polymers in the circulation is a biomarker of your disease. We had a subject with antitrypsin deficiency who had a liver transplant, and we could see the polymers fall. So here's a point of liver transplant, and after the transplant, the antitrypsin levels go up, as you'd expect. And here's the polymers that are in the circulation, and they go away and become undetectable by about day 5, showing that the polymers come from the liver and that they clear following liver transplantation. So understanding the process of polymerization also allows us to follow the clearance, and we think that the clearance of these polymers is about 30 hours. Polymers in the liver, polymers in the circulation, and we believe that polymers are also important in the associated lung disease. So if you lavage individuals with antitrypsin deficiency, we can find polymers. And as Adam said, Manuel Cosio presented at this meeting macrophages in the lungs of individuals with antitrypsin deficiency that are full of polymers. So these polymers are in the lungs, and in the lungs we think they're inflammatory. We think they cause an influx of neutrophils. So what do I think is happening? I think that you've got polymers in the liver, so the material doesn't get out. The limited amount that gets out gets into the lung. If you smoke, it drives the protein to form polymers. Those polymers don't work. They don't function to inhibit enzymes, and they're also inflammatory. They recruit neutrophils into the lung, and hence we have this development of emphysema. So the emphysema is more complex, at least to my mind, than just a deficiency of a protease inhibitor. It's also got this component caused by polymers that drive inflammation and make the disease worse. And of course, the most important interaction is with cigarette smoke. So now we understand the mechanism, can we use that to drive therapeutic innovations and therapeutic strategies? Well, the therapy that you'll all know is antitrypsin replacement therapy, and we can debate efficacy and who you treat, how often you treat, how effective it is, and the benefits. Even if it works phenomenally effective, and there's some debate about that, then it won't treat the liver disease. It'll only be effective in individuals with associated emphysema. So what we've tried to do is to say, let's see whether we can develop small molecules that block the proteins linking together, and hence block the fundamental process of this disease, the formation of polymers. So here again is my model. Here's the antitrypsin molecule. It opens up, and as it opens, we have significant movement. Another molecule links here, and hence we have this chain of polymers. So what we're trying to do is to use a small molecule to block two proteins binding together. and we're trying to do it in the middle of the liver, in the cells of the liver. It's a big ask. That's a big challenge for anyone to try and do that. So let me tell you how we approached it. So here again is the antitrypsin molecule, and normally it's nicely closed, looking like that. And what the mutation does, what the Z mutation does, is to make it look like that. So it opens up the molecule. It opens up the heart of the molecule, and it allows another strand for another molecule to insert, and hence to form this dimer, and hence chains of polymers. I want you just to point out that in this part of the molecule, there is significant movement as we go from the closed to the open configuration. Can you buy that? You can see that in order for a polymer to form, this molecule has to open, and there's significant movement here. So when we looked at our crystal structures of the protein, we could find that there's a cavity here, there's a space. And very simply, if we can put a small molecule in this space, if we can stop this protein opening, then we overcome the effect of the mutation, we keep these 3 yellow strands in place, we stop another strand inserting, and we will prevent disease. And that was our starting point. So I went around the pharmaceutical companies and said, I've got a terrific idea, and they said, that's very clever. Very clever, David, very clever, but that's really hard. That's never going to work. And I knocked on many doors and had many people say, there's no way forward. And so what I did is to team up with a group at the Scripps Institute and say, okay, can we find small molecules that will bind to this pocket to see whether we can actually find just proof of principle that this could work? Because if I can get to that point, I can persuade a pharmaceutical company to be interested in what we're doing. So the Scripps were very interested. They screened 1.2 million compounds in silico, so on a computer, to find out ones that could potentially fit into this pocket and prevent the molecule opening. They found 68 potential hits, and then we bought them commercially. We couldn't have them made. We're not that type of research group, so we bought them off the shelf, and we said, will they work? Will they block polymerization? So this work was done by a fellow, a medical fellow in the laboratory, who was given the task of naming the molecules, working with them in order to see which ones were effective. So instead of calling them 1 to 68, which seemed the most logical way of doing this, he named them after the only thing in the world that he understood. He came from North London, he supported Arsenal, and so he named them after the London Underground. And he named each molecule after a tube station on the London Underground. He named each derivative that we made after a monument next to a tube station on the London Underground. And each derivative of a derivative he named after a road, next to a monument, next to a tube station. And so for 18 months, this wretched map was the focus of our laboratory meetings. The first molecule that came out of the screen and was shown to be effective was called CG. Anyone from London? Covent Garden, terrific, thank you. Well done, Covent Garden. So the first hit was Covent Garden, and when he left the lab, we bought him a picture of Covent Garden, okay, as a souvenir of his time in research. So this is plasma isolated from anyone in this audience who's a Z homozygote. We have a clinic, we'll regularly take blood from people and then purify the protein. That's what it looks like. If we incubate it under physiological conditions, it forms polymers. This is what the polymers look like. It's a ladder on a gel. So this is what the polymers look like. All the small molecules were dissolved in a solvent that's called DMSO, and hence this is the positive control. And for the molecules to work, they have to block this chain of polymers. Here's 6 compounds. Here's Covent Garden, compound 3. Compare that lane to that lane, and we've abolished polymerization. Okay. Phenomenal. We were delighted with that. So then we went back to Scripps and said, terrific, we've got a few that work. And so they took Covent Garden, they bound it into the pocket that we were interested in looking at, they relaxed the pocket around the molecule, and then we rescreened 1.2 million molecules looking for other binders. Okay, so we're looking for things that will fill that pocket and block polymerization. We had another 19 potential hits, of which 10 block polymerization. Here they are. Here's the starting material. Here's the polymers. Here's all the successful hits. And just to help you with the nomenclature, remember they're all derived from Covent Garden, and so those of you who know London will know the Covent Garden region. Longacre, English National Opera, Monmouth Street, Seven Dials, Theatre Royal and Westminster Hall. And you think I'm joking. If you go to the paper, which is published on this, it actually shows the very chemical structures and the derivatives, and all the letters are shown in that paper. And when it was accepted, the editor said, could you change these letters to numbers? And we said, no. We'd lived with these letters for 18 months. And so as a consequence, I think it's figure 1D in the paper has these various letters, and only a few of us know that CG is not some terrible chemical structure, but is actually Covent Garden. So what we have then is a potential starting point for drug discovery. And if we look at the pocket in more detail, you can see it's got a head, a body, it's got a tail, and you can modify each of these to try and get better binders to try and improve polymerization. So I went to GSK, who I was then working with quite closely. I chair their therapy area board, which sort of gave me a way in, really, in terms of saying that antitrypsin deficiency really was an area they should focus on. I sort of persuaded them that was the way forward. And so 5 or 6 years ago, we developed a partnership between GSK and academia to do this properly. Because what I've told you about is a bunch of academics trying to do drug discovery. This is the way of doing it properly. So we've worked with them for a long time. I obviously can't show you the data, because I can't do that, but all I can say is that 5 years later, we're still going. It's a tricky process. The medicinal chemists that I work with describe it a bit like, do you remember the James Bond film? Okay, Bond film where James Bond ran across a series of alligators in Live and Let Die. There's a few of us of a certain age that remember that, and I think it was Roger Moore. It was James Bond in the Roger Moore incarnation. I think it was 1973, I think. So James Bond runs across a series of alligators to escape from the baddie, from the island back to the shore. And the medicinal chemists at GSK said to me, David, what you need to understand is that in drug discovery, if the first alligator doesn't get you, the second one will. If the second doesn't, it'll be the third and the fourth and so on. So where are we? Well, I think there's about 10 alligators across the river, and we're probably about 5 alligators in. So does that mean we'll get that? Dunno. But at least we've had a really good try, and we'll go down fighting. More recently, we've taken a different approach, and we've made antibodies that will block polymerization. Here's one of our antibodies that we made with funding from the foundation, actually. And this antibody, here's polymers, and with the antibody, we can completely abolish polymerization. And then when we put that into liver cells or models in the cell, then we can actually increase the amount, we can reduce the amount of polymers, compare that with that, and we can get the polymers out of the cell, compare this control with that. So we think this will work. We think that a strategy to block polymerization is likely to be effective in this condition. So to summarize what has now been 25 years of work on your disease, that we think that antitrypsin deficiency is a novel protein-protein interaction that we've called loop sheet polymerization. And for me, this is all about polymers. Understanding polymers gives us new strategies to treat this disease. And for me, the cure is a small molecule that blocks the protein-protein linkage in the liver and releases normal material into the circulation to protect the lungs. And finally, I'd just like to thank the people who funded all this work. The Medical Research Council in the UK have funded it now for 25 years. Welcome, Alpha One Foundation, and of course GSK, who ultimately, if we get there with a cure, will be the most important partner in this. Thank you very much.